Topic: culturing. At the end of the lesson, students should be able to define the term culturing, list 5 instruments used for the preparation of culture solution, outline the process of culturing microorganisms in the laboratory, state precautions that must be taken during the preparation of culture solution in the laboratory. Definition: culturing is a technique of growing microorganisms in special media in the laboratory. The major instruments required for the preparation of culture solution are inoculating loop, inoculating chamber, cover slips, slide, petri dish, test tube holder, test tube rack, and lens, and microscope. Simplified diagrams of the instrument that I've mentioned earlier include the test tube holder, the test tube rack, the agar nutrient that is used to grow microorganisms in the laboratory, the petri dish, the Bunsen burner, the hand lens, and the microscope that is used to magnify the colonies that cannot be seen clearly. But the microscope will make it possible to see the colonies of microorganisms growing in an agar nutrient within a petri dish. Preparation of culture solution. The first step is to boil the culture solution, agar, and pour it into the sterile petri dish. Allow it to cool and set in the petri dish. Introduce the material, dirt from under the fingernails, contaminated food, contaminated water, or you expose the petri dish to air. These ingredients are allowed into the agar medium with a sterile inoculating wire and cover immediately. Place the petri dish in warm but dark compartment or an incubator. Observe and record for 2 to 3 days. Colonies of microorganisms grows in the culture medium. Microorganism grows gradually until the cultural solution is completely consumed by developing colonies of microorganisms. You can see the petri dish, the inoculating loop, and the colonies of bacteria developing on the agar jelly. Pick up the agar nutrient, boil the agar nutrient, pour it into a sterile petri dish, sterilize the inoculating loop, pick contaminated food materials with sterilized inoculating loop, streak the agar in the petri dish, cover the petri dish immediately and seal it with a sellotape. Seal petri dish with sellotape is placed upside down in the incubator to enhance microbial growth. The growth is allowed to take place at a temperature range of 25 degrees Celsius, after which the plate is removed and viewed. View bacterial growth clearly via a microscope. Precautions observed during culturing. Wash hands with soap before and after the preparation of agar solution. Open petri dish only slightly and cover at once. Close petri dish firmly with adhesive tape. Sick persons should not be permitted to take part in the culture experiment. Avoid talking, coughing, sneezing, and touching of infected jelly. Unused agar should be sterilized by washing with antiseptic soap and disinfectant. Example, 40% formaldehyde. It can also be used to sterilize. All instruments should also be sterilized before the beginning of culture solution preparation and after the experiment. Wash hands with soap. Open petri dish only slightly and cover at once to avoid contamination. Close petri dish firmly with adhesive tape. The following precautions must be taken into consideration while preparing microorganisms in the lab. A lab coat should be worn to protect the clothing. Hands washed before and after experiment to remove microorganisms. Smooth non-absorbent work surface. Clean with disinfectant before and after experiment to kill microbes. Autoclaves or pressure cooker is used to sterilize all glassware. Sterile petri dishes of nutrient agar kept closed until required to prevent entry of contaminants. Wire loop flame before use to destroy unwanted microbes. Lid held over petri dish during inoculation to prevent entry of contaminants. Inoculated dish sealed with tape to prevent entry and exit of microbes. Bin lined with plastic bags for the safe disposal of used plates. Thank you.